Good afternoon and welcome to the fourth scientific webinar organized by GFB Onlus. First of all, I wanted to thank you for the trust you have shown. During all the webinars done, we have touched more than 50 assistants and more than 150 views on YouTube registered talks. Thus, in name of GFB, thank you. As you well know, our association was born 10 years ago, and since then, it has always supported research and understand and find treatments for the different forms of limb-girdle muscular dystrophy, with special interest in the 4 subtypes of sarcoglycanopathy. That's why today we are particularly pleased to present the research of Professor Dorianna Sandonà that have been working in sarcoglycanopathies for a long time. Professor Sandonà has had the degree in biological science for almost 30 years at the University of Padua. After several years of research in various laboratories, Professor Sandonà returns to Padua, where she coordinates a research group combined with teaching activities. One of the hot topics of her research activity is the degradation process of proteins with folding defects, which play an important role in muscular dystrophies such as sarcoglycanopathies, but also in other rare genetic diseases. The objective of Professor Sandonà's work is precisely to identify common molecular mechanisms in different pathologies in order to be able to apply similar therapeutic strategies. In particular, her group studied small molecules called correctors used for the treatment of cystic fibrosis that have also proved to be useful in sarcoglycanopathy. However, there are several issues that still need to be addressed, such as how they work, how efficient they are, what are their side effects, can they be used for different forms of sarcoglycanopathy, and moreover. These are the questions that we will discuss with Professor Dorianna Sandonà. Professor, the floor is yours.
Thank you very much, Carlos, for this beautiful introduction. And if I try to share the screen.
Yeah.
Okay. Tell me if it is working.
Yes, now it's working.
Now I try to put in full screen. Okay. Is it working?
Yes.
Okay. So
Perfect.
first of all, I would like to thank not only Carl for this introduction, but also the entire GFB Onlus, because first of all, for these really exciting webinars related to sarcoglycanopathy, and not only, but in particularly, I would like to thank the president, Beatrice Vola, because she was one of my first supporter, considering our work. And this said that, I would like to go on. First of all, this is my disclosure, because some of data are protected by patents. And few additional thanks, because I would like to thank my collaborators. This is a small group, but is composed by really interesting subjects. And among them, I would like to mention particularly Martina Scano, Marcello Carotti, and Alberto Benettollo, because they did most of the data that you will see during the talk. But there are several other components that are not strictly in my group, but they are strict collaboration, such as Roberta Sacchetto, Blaubert, Leonardo. These guys were very useful. They helped a lot for the experiment of functional activity of muscle, and also Sofia Parrasia, because she helped us in the pharmacokinetic studies. Of course, I would like to thank Fondazione Teleton, AFM, Muscular Dystrophy Association, as well as my university and the Ministero dello Sviluppo Economico from Italia, because they funded part of this work. And last but not least, again, I would like to thank the Cystic Fibrosis Foundation, as you will see in a while, and the GFB. But let's go into the story. As I told you, we will focus specifically on sarcoglycanopathies that you well known are part of the big family of limb-girdle muscular dystrophies. Because of the previous really interesting webinar, and because I know that most of you well know these diseases, I don't want to enter in detail into the clinical aspect and so on, but I would like just to mention a few things, because sarcoglycanopathy is in the reality are 4 diseases, because there are 4 genes coding for 4 proteins that are alpha sarcoglycan, beta sarcoglycan, delta, and gamma sarcoglycan. They are acting in our skeletal muscle like a unit. They are together, and when we have defect on one of these genes, and so in one of these protein, we lose the entire complex. Sarcoglycanopathy are rare diseases, and by the way, the sarcoglycanopathy in the large family of limb-girdle muscular dystrophies account for quite a lot of forms, because they are about the 15–20% of all LGMD. And by the way, they are among the most severe. I don't want to explain in detail the clinical features of sarcoglycanopathy, because again, you well know this, but I want to mention that the age of onset of the disease could be different, could be variable, but very often it occurs very early into the childhood. The subject with sarcoglycanopathies very often lost the ambulation during the adolescence. There are several other consequences due to the muscle wasting, such as contractures and scoliosis. Another very common mark of the disease is the high level of the creatine kinase into the bloodstream, and this is because the damage of the muscle that release this specific enzyme. We can have respiratory problems, cardiac problems. These are much more depending on the subtypes of the sarcoglycanopathy, and never was observed cognitive impairment. A very important point is that sarcoglycanopathy are still without a cure. We well know, however, that there are some possible therapeutic strategy that can help in reducing the symptoms and in maintaining the activity, the locomotion activity, the daily life better as long as possible. And these therapeutic approaches are possible because of the use of corticosteroids to reduce inflammation, physiotherapy, surgical intervention, respiratory support, and also the care monitoring of cardiac function and support. However, we know that these are symptomatic treatments. In other words, they are helping the state of the patient, but they are not curing the disease. However, we know that sarcoglycanopathies are genetic diseases. And by the way, the genes are also quite small. And so you well know that the most effective approach is expected to be the gene transfer therapy. We have a problem with the gene, we can try to add a novel, correct, healthy copy of the gene to cure the disease. This is effectively a causative treatment. We are curing the disease. I don't want to enter in detail on this, also because you had the last webinar by Dr. Richard that exactly was on this topic. However, I would like to tell you that sarcoglycanopathy are indeed genetic disease, and there are a number of different types of mutation that are the cause of the disease. And this mutation could be large, deletion, insertion. So they result in this case in the complete absence of the protein, of the sarcoglycan. But we can also have small mutation, and small mutation can have different outcomes, because sometimes this small mutation could affect the structure of the protein in terms that they alter completely the structure of the protein, the frame shift mutation. All this, from the point of the mutation, the protein is completely different. Or the null mutation induce a premature stop of the protein. So the protein is very short or truncated. In this case, we have the disruption of the protein. But there are another part of the proteins, of the mutation, that result in a protein that is called defective sarcoglycan. These are mutations that change a little bit in the protein, but do not alter completely the sequence of the protein. And by the way, as you can see from this graph, these type of mutation are quite a lot. They account about 60–65% of all the mutations overall, considering all the sarcoglycans. But the point is, which is the outcome of this different type of mutation? So this is the situation that we can observe in a healthy subject, in the muscle of a healthy subject. As you can see here, this is the histological analysis of a piece of muscle. And here you can see that the fibers, so the component of the muscle, the cells of the muscle, are very irregular. And for what is important for us, concerning the sarcoglycan, you can observe through an assay that is called immunostaining, we can visualize at the level of the muscle the presence of the different sarcoglycan, the presence and also the localization. And as you can see, they are indeed at the border, on the surface, on the membrane of the skeletal muscle cell. And here, the sarcoglycan complex, they are all together, plays a very important role in assuring the strength of the plasma membrane, the sarcolemma. And so, in this manner, the muscle can sustain the stresses due to the muscle contraction. But in sarcoglycanopathy, what happens is a very different things. Because as you can see here, in sarcoglycanopathy, we observe, first of all, that the skeletal muscle is damaged. you can see that there is not a regular shape of the fibers. They are different in size, and there are also all these small blue spots that are cells of the immune system that induce inflammation at the level of the skeletal muscle. And furthermore, you can also observe this region of pale pink that are region in which the contractile tissue is replaced with stick tissues. It's something that is unable to contract, that is the fibrotic tissue. And what about sarcoglycans? As you can see, you don't see anymore the signal of the sarcoglycan. No one is there. And this is the situation in which we lose the sarcoglycan complex. Again, the mutation that can lead to this are the large gene deletion insertion, or can be those that disrupt the structure of the protein of the sarcoglycan, so the frame shift, the null mutation. But as you can see here, there are other situations in which we have the same problem at the level of the muscle, but you can observe that there are some traces of the sarcoglycan proteins. And so in this case, we are in a situation like this. We have a strong reduction, but some is still there. And these are very often the mutation that leads to the defective sarcoglycan. So at this point, the question is, but what is a defective protein? So I prepared this very basic cartoon to try to explain what does it mean to have a defective protein, a defective sarcoglycan. So suppose that this is our proteins, our sarcoglycan. This is Lego. Each one of these pieces are the amino acid of our protein, the constituent of our protein, and they form this long chain. But in this shape, the protein is not functional, is not active, because the protein needs to assume a form, a shape, a configuration, a structure. Only when the protein is able to gain this structure is active, is function. Unfortunately, when we have a small mutation, like the one that I showed you before, this is really a very small problem at the level of the chain. But this can result in a protein that is unable to gain the proper folding, the proper structure. So this is the defective protein. And once the cells sense that there is this kind of problem, not always, but very often can activate a system that leads to the degradation of the protein, because the cells thinks that if I have this defective protein, this could be dangerous, and therefore it's better to destroy it. But how is destroyed? How are a sarcoglycan protein with this kind of defect be destroyed? Again, one step back to think the normal growth of a normal sarcoglycan. They are synthesized inside a particular organelle of our cell, that is the endoplasmic reticulum. Here, sarcoglycans undergo synthesis, folding, they are modified by glycosylation, for example, and at the end, they are able to gain the native structure. And so, once all the 4 sarcoglycans are ready, they can form the complex and can leave the endoplasmic reticulum, going to the surface of the skeletal muscle cell, so reaching the sarcolemma. What happens when we have a defective sarcoglycan? So in this case, we have a very small mutation. Do you remember the orange element? This allows the cells to sense that there is a problem. And so the endoplasmic reticulum activate a very complex mechanism that is called endoplasmic reticulum associated degradation, that I don't want to explain in detail, but you have to know that once this occurs, the protein is recognized, is removed from the endoplasmic reticulum. It is marked with a stamp with the molecule of ubiquitin, and this is a signal for being degraded through the proteasome. So at the end, what happens is that if most of the protein take this route, a very few of the protein is able to fold in the more or less proper way and to be able to form the complex with its partners, reaching the sarcolemma. So the point is it possible to overcome this problem, to revert this route in order to improve, to help the endogenous sarcoglycan that is mutated to form the complex and to reach the sarcolemma, even though containing the mutation? How to do this? We have an idea that was coming thanks to the knowledge of another disease, that is cystic fibrosis. You know that cystic fibrosis is a very severe disease, is the most common lethal disease among the Caucasian population. It's a complex disease affecting several organs. I don't want to enter in the detail. Another feature of cystic fibrosis is that the protein that is called CFTR, that is responsible for the disease, present a huge number of different mutations, 1,000. And however, among these huge number of mutations, there are some of them that are called type 2 mutation, mutants, that has exactly this story, that follow exactly this story. They are small mutation, induce the production of a defective CFTR and are degraded. Because of the frequency of this rare disease that is not a very rare disease, there is a lot of study about cystic fibrosis, and this allows us to identify also a huge number of small molecules that have been developed to treat pharmacologically this disease. And among these small molecules, some of them are called correctors because they are able to correct the folding defective CFTR. So if this is the protein with the mutation, by the addition of this small molecule, the protein is able to gain a structure that is very close to the normal CFTR corrector. They are called indeed CFTR correctors because they correct, they help the protein to gain a structure that is similar to the normal one. And so our idea was, it's possible to use these small molecule, these CFTR correctors for the pharmacological treatment of sarcoglycanopathy. Of course, we can just focus on the defective sarcoglycan, but also in this case, we should have a sort of causative treatment. So we are trying to cure the disease, acting on the endogenous mutated sarcoglycan. So acting to recover, to help, to correct the mutated sarcoglycan. Thanks to the Cystic Fibrosis Foundation, we had the chance to have a small amount of several of these CFTR correctors. This is the list that we use, but of few of them, we had this small amount, and we try to test these compounds in sarcoglycanopathy. How to do this? First of all, we started with a cell model. Human cells, these cells are commonly used in research because they are really very versatile. You can introduce in this cell, for example, alpha-sarcoglycan. And if we introduce in this cell alpha-sarcoglycan wild type, so the healthy sarcoglycan, we exactly observe what is present in muscle. So the sarcoglycan is able to reach the surface of the protein, as you can observe here. On the contrary, if these cells are transfected, so produce a mutated form of alpha-sarcoglycan with this small mutation, this change, this is the orange element that you see in the protein, you can observe that our protein is no more able to reach the plasma membrane, is destroyed. However, if we treat for 24 hours with different CFTR correctors, you can observe that we have again the presence of the sarcoglycan at the level of the plasma membrane, like in the wild type, even though the protein is mutated. We were happy, of course, about this, and this allows us to go on in the story, searching, first of all, to a model in vitro that is closer to the real life, to the skeletal muscle. And again, to do this, we had the possibility of using a very useful and things that is myogenic cells, so cells that are coming from patients with sarcoglycanopathy. And we have to thank for this the Italian Teleton Foundation biobanks that store a huge number of these precious material that is coming from biopsies of patients. And we have also to thank Marina Fanin, that is no more with us, but is still in our heart, because she helped us to put in culture these cells, to grow these cells, and also to differentiate these cells in order to obtain the myotubes that are the first step of muscle differentiation. And so in these myotubes, we use CFTR correctors. Here the story. So here are the myotubes, as I explained. These are myotubes from a patient with alpha-sarcoglycanopathy. This is a compound heterozygote because present two different mutations on the two sarcoglycan allele. And you can observe here, you can appreciate here that the amount of alpha-sarcoglycan, delta-sarcoglycan, remember, here we are in the real cells. And so we have all the sarcoglycans. One is mutated, the alpha, the other are the normal one. Nevertheless, you see that if we don't do nothing, so we treat just with the vehicle, we see just a faint signal of the sarcoglycan. So we are in this situation. But if we add the CFTR correctors, you can observe that the signal of alpha, delta, beta sarcoglycan is very much higher at the level of the sarcolemma. So we are in this situation. This was really exciting. And the second thing that we did was to try to do the same with beta sarcoglycanopathy cells. In this case, again, thanks to the biobank of Teleton, we had these cells that are from a patient with this kind of mutation. This, in this case, is an homozygote. And again, we observe that we are able to increase the amount of the protein after the treatment. And most importantly, we are again able to see the protein at the level of the sarcolemma. Nothing or almost nothing before present after the treatment. And so also in this case, we have this situation with the sarcoglycan complex in place with a subunit that present a mutation. Because of this efficacy in alpha and beta, we decided to identify our C17 as our best corrector, the one that is able to act on different, that has a broad activity. However, remember that we have this recovery. The sarcoglycan is on the sarcolemma, but one subunit is mutated. And so the point is that we need to understand if the presence of this complex containing the mutated sarcoglycan is able to do the job, is able to preserve the structure of the sarcolemma, is able to avoid damage at the level of the recovered muscle. You can understand that this is quite difficult to do in vitro, because it is difficult to allow cells, for example, to contract. We need to pass in vivo. And this was a quite difficult task at the beginning because of the lack of suitable animal model. Because indeed, of course, we have the wild type animal, mouse, and here is just to show you that the situation is exactly like the human. We have a regular organization of the skeletal muscle. We have the sarcoglycans on the surface of the skeletal muscle fiber. And by the way, this animal is also able to do the muscle movement. The strength is normal. We are in this situation. There is another very well animal model that is the knockout. And the knockout is an animal that presents all the signs of the disease, because look at the muscle histology, there is no sign of sarcoglycan complex, so we are in this situation. And of course, we have poor muscle strength, so we have damaged muscle. But as you can understand, this animal that was exceptionally useful for testing gene therapy, because this is an animal in which there is no alpha sarcoglycan, so you can introduce again alpha sarcoglycan in the correct form, and you can recover the problem. But this is not useful for us, because we need an endogenous, we need the presence of the endogenous alpha sarcoglycan with the mutation. And so we try to solve this problem with inducing the expression of an alpha sarcoglycan mutated, folding defective, in the background of the alpha sarcoglycan knockout. And so we take this animal, newborn, and we injected just in the hind limbs the sequence of the human alpha sarcoglycan with this particular mutation. We wait 7 weeks. In this time, during this time, the animal grow and develop, and at the end, we have an animal with humanized hind limbs that express the alpha sarcoglycan with the mutation together with the endogenous beta, gamma, and delta sarcoglycan. This animal was analyzed, and of course, you can observe that there is a feature of dystrophy. There are traces of the different sarcoglycans, so we are in this condition, and there is poor muscle strength. So this is an animal presenting a defective alpha-sarcoglycan, useful to test our approach. And indeed, we did a lot of experiments with this animal. Here are reported the different regimens that we adopted. We did chronic treatment with the C17. This is the dose that we used. We injected the compound intraperitoneally, and we perform experiment long 3 weeks with daily injection, 5 weeks with daily injection, and 5 weeks with injection every 2 days. And I would like to show you data coming from this last experiment. Oh, sorry. So first of all, we analyzed the muscle of this animal. And as you can see here, that if we don't do nothing, so this is just the vehicle, we introduce the vehicle, you can observe that there are traces of the different sarcoglycans, but nothing else. We are in this condition. However, if we treat with the C17, you can observe that again we have the alpha-sarcoglycan, that is the mutated one, on the surface, together with delta, but also beta and gamma sarcoglycan. So we are in this situation. Very happy. The last thing that we have to evaluate is if this condition, so this rescued condition, protect muscle fibers from damage, allowing the muscle to work properly, to have force. And so we evaluated the activity of the muscle mouse in vivo. Of course, we analyze some control, so the healthy wild type mouse, the knockout mouse, and then the humanized mouse treated with vehicle or C17. This is a curve that show you that if you excite, if you stimulate the muscle of this animal with different frequency, you can observe that at the end you don't see any movement, but then you can the muscle can move and can move a weight. So it produces force. And you can see that this is what at the different frequency of stimulation do the wild type mice. This is what happens with the alpha-sarcoglycan knockout, a very reduced force. But what happens if we use the same protocol with the humanized mouse treated with vehicle? The situation is very much similar to the one of the alpha-sarcoglycan knockout. So the two curves, as you can see, are superimposable. And what about when we treat the humanized mice with the C17? The answer is very well, because you can see that this animal present a higher force in comparison to the negative control. So we can say that both alpha knockout and humanized plus vehicle have a poor muscle force, whereas the humanized treated with C17 recover the force in a similar way like the wild type. The next slide show you exactly the same thing, but focus just on one specific stimulation frequency, that is this one, and the result is visualized in a different way, but again can show you that the knockout and the vehicle treated are pretty the same, and the C17 are much more similar to the wild type. So we can say that the proof of concept of our treatment have been obtained. And so we are happy, but we cannot stop here. We have to go on with additional work. And so one of the first things that we tried to do was to understand if C17 is safe or present any toxicity. We take advantage from the data that we obtain with the chronic treatment. So remember that we treated for 5 weeks every day or every 2 days the animal. And from this experiment, we can say that we observe no acute adverse event. All treated animals reached the experimental endpoints. We observe no reaction, allergic reaction, for example, at the site of injection. We observe no effect on the growth, on the appearance, and on the behavior of the mice. Here is just the body weight of the animal at the end of the treatment. And you can see that they are similar, identical between vehicle treated and C17 treated. We observe no macro or micro tissue damage at the level of liver, kidney, heart. These have been already evaluated. Here is liver and kidney. You can observe that there is no difference. And other organs are now under evaluation. We observe normal renal filtration, so there is no problem at the renal level. We evaluated the red blood cells, the white blood cell, the platelets, and we observed that they are normal, in the normal range, as well as the hemoglobin content was the same. So this can tell us that even though these experiments were not according to the rule of the FDA, of course, we did in our laboratory, but they suggest that there is a reasonable safety profile of C17. The other point that is essential to do is to understand if C17 is able to reach the proper tissues and if it is eliminated at the end, because if we have the accumulation, this could be detrimental at the end. And so we did a preliminary experiment of evaluating absorption, distribution, metabolism, and elimination in mice. We use exactly the same route of administration, so intraperitoneal with the same dose, but in this case we did an acute administration, just one administration, and then at different time points we collected blood and as many as tissue as possible, and from these tissues we analyzed the content of C17. I cannot show you exactly the data, but I can tell you that these preliminary experiments were successful because we observed that C17 is normally absorbed. Of course, it reached at first liver, kidneys, and then it is redistributed to the bloodstream to the other tissues and organs. It reaches the brain, the heart, the skeletal muscle, where the maximum peak is at 2 hours, but then is still stable up to 6 hours. Then start to reduce the amount, and after 48 hours, it's ranging from 0 in certain tissues to 6.5 in skeletal muscle. It is removed from the organism through the urine and to the feces, modified in order to render the molecule hydrophilic from the urine, and both modify and modify in the feces. And we have also first idea that the C17 is metabolized at the level of the intestine, but we have a lot of work to do on this, because first of all, we are working on mice. That is not the best choice for this kind of experiment. We should use other animals such as rat, for example, but this is really interesting. So this is our overall preclinical timeline. The timeline is not in scale, as you can see here, but we can see that in these first 10 years, we did a lot of work from the discovery, from the idea, the screening, the validation in vitro and in vivo, the efficacy of the compounds, the first pharmacokinetic studies, toxicology, and now from this point we are studying, we want to study the delivery because we want to understand if it's better to use the oral or the parenteral administration. We would like to also to possibly optimize the lead compound, the C17 molecule, because we can improve the delivery or at the muscle cells, for example, and so on. And furthermore, we have to do all these pharmacokinetic and toxicology studies in the so-called hind enabling method, by using the FDA rules in such a way at the end we are ready to start with the clinical trial. So to conclude, we can say that we are proposing the novel use of a small molecule as a pharmacological therapy for sarcoglycanopathies that are caused by mutation resulting in folding defective sarcoglycan. So remember that we are focusing on this part of this graph. We have solid data of efficacy. I think I hope to have convinced you about this, obtained both in cell model, but in cell from patient and in vivo using this humanized mouse model. We have a pharmacokinetic and preliminary toxicity analysis that, even though are not performed according to the FDA rule, are suggestive of reasonable drug-like properties and safety profile of C17. C17 is a small molecule, so remember that can be easily synthesized, optimized, and administered and delivered. So we believe that C17 is a good candidate to be further developed. Last, mutation leading to folding defective sarcoglycan account for the majority of the sarcoglycanopathy cases. So we have a large group of subjects that could, in principle, benefit from this strategy if it will go on in the development. Furthermore, the rescue of just one allele is expected to be sufficient to ameliorate the phenotype, because the disease is autosomal recessive. So if, in principle, if we have one allele that is unrecoverable because of the particular mutation, but the other one is possible to recover, this should be enough. And remember that we have a quite high level of compound heterozygosis in sarcoglycanopathy. And finally, our pharmacological strategy could be considered complementary to gene therapy. They are not mutually exclusive. In principle, we can have that the pharmacological strategy could be adopted in case, for example, it's impossible to use the gene therapy for any reason because of the presence of antibody against the adeno-associated virus or something like that, and vice versa. If should our strategy fail, the subject could be eligible for gene therapy treatment. And finally, and this is another interesting point, it's possible to do combo treatment because, for example, we can use something for reduce inflammation and at the same time, we can try to rescue alpha-sarcoglycan. And so this is really the last one, and I would like to thank you very much for the attention.
Thank you, Professor Sandonà, and very nice presentation and very clear. So I hope the audience understand very well because it was very clear. And now it's a turn for the questions. We have already received several questions from all over the world because I remember that these webinars are for our international audience. So we have people hearing from the US, from Russia, from everywhere. So now I start some of the questions. Maybe you already answer or start to answer, but even if we can repeat it, it's better.
Yeah, sure.
Just to let it clear. So the first question is about the missense mutation. They say that there are a lot of missense mutation. There are quite many, and they wonder if will be all missense mutations to be rescued for this treatment, or just some of them. So how to determine which missense mutation could be rescued for these small molecules?
Okay, first of all, it's true there are a large number of missense mutation. We didn't test all of them. We test some of them, but we have, with the ones that we have tested, we have good results because all of them were rescuable. Just one was less rescuable, but in this case, by using a combination of CFTR correctors or even CFTR correctors and other small molecule, is possible to recover them. So, of course, I cannot say now, yes, all mutations are rescuable, but many of them, yes. And furthermore, in thinking to the personalized medicine, by the possibility of using cells coming from the patient, we can easily understand if they are recoverable or not, as I show you with the alpha-sarcoglycan patient, with the beta-sarcoglycan patient. Mm-hmm. So this could be quite easy to do.
Okay, very nice. So now another question that you already mentioned, but they made it from different places. It's about the combination of the treatments. So when this drug will be a reality, could be administered in combination with gene therapy. No, I mean not or gene therapy or this drug. I mean the combination of both of them.
Okay.
Or patients should should try one on another.
Okay. No, allora, gene therapy, as I told you, is for sure the elective therapy when you don't have any sarcoglycan amount inside the cell because the mutation destroyed the gene or is so strong in the structure of the sarcoglycan, so null mutation, out of frame mutation. In this case, our system is not working. We need that there is the protein that can be formed, even though containing the mutation. Okay, so I think that it's not useful to do gene therapy together these, but suppose that you have this kind of mutation and gene therapy is not effective because, for example, there are antibodies against the adeno-associated virus, so the delivery vector, okay? But you have the mutation that is possible to be rescued, maybe it's better to use, if it will reach the final point, our strategy. On the other hand, if the pharmacological treatment failed for some reason, the patient in any case could have access to the gene therapy approach. Okay.
Okay. Yeah. So you can get your the treatment of a small corrector, and if it not works, you can get.
You can do the exactly.
Okay, it's clear. Another question: if this drug works in LGMD patients, will be easier to get approved for the usage since the cystic fibrosis medications are approved?
Okay, this is a very essential question because the molecules that we are using, this C17, just to give the name, is not one of the CFTR correctors approved for cystic fibrosis. And this is the reason why we are doing all the experiment to check the toxicity, pharmacokinetic, and so on. So the timing is could be slightly accelerated because we have many information coming from the cystic fibrosis story, but we have to do all the steps in any case.
Okay. Well, so another question, it's how many times per year could be these therapies administered to a patient? I mean, and for how many years? And then could you explain how it is administered to the patients with cystic fibrosis? And for the LGMD patients, should be something similar? Exactly,
this is the point. Should be very similar to the cystic fibrosis, because in this case, we are not curing for with just one administration. We don't have the recovery of a protein that will be there for every time. This is a chronic treatment. You need to take the pills or what else every day, like in cystic fibrosis. They assume the CFTR correctors twice a day. But what we are thinking to do is the delivery method. So the administration could be a pill, but could also be something, a system that can be placed under the skin and that can release the compound very slow. We are thinking all these things to have the better compliance for patient, of course. Okay. So another question is, how does it cost to the national healthcare system, the drug administration in the cystic fibrosis patients? To know, yes. For example, we know that gene therapy is very, very expensive. And yes. Okay. In this moment, I don't remember. I had this data because I know how much it does cost cystic fibrosis treatment. But for sure is several order less than gene therapy, even though is expensive. It's not cheap, but is less expensive, much more less expensive than gene therapy, of course. If you want, I can write in the chat later on this cost, because I had this information, but I don't remember. I don't want to tell you data.
Okay, okay. No, if you write it, maybe then if the people that make this question are interested, we can give the answer to them. So yes. And another question coming from the States is that there is any pharma industry developing the drug for say, cystic fibrosis, and that should be, and that pharma should be interested in developing drug for sarcoglycanopathies.
This is a very good point. Yes, of course, Vertex Pharmaceutical are those that are developing the CFTR correctors for cystic fibrosis. And we are trying to get in contact with them to try to ask if they are interested in developing CFTR also for sarcoglycanopathy, for example. But until now, we don't have this
answer. But what's the name? Sorry that I
Vertex Pharmaceutics.
Okay, okay, okay. And another question, and I'm arriving to the end. And there is a similar therapy in France developed by Dr. Isabelle Richard. It is the same, or
no, it's not. No, it's not the same, or better. We are collaborating because we have grant together for the CFTR correctors, but Isabelle Richard is also studying a different compounds that are acting either like CFTR correctors, but they are different, so the mechanism should be similar, even though the molecules are different. And she was also involved several years ago in checking molecules that are acting in the degradation pathway. So blocking the degradation of the CFTR correctors. She published, like us, by the way, several years ago, papers showing that if you block by using inhibitors of the molecule that recognize the folding defective protein, or if you block the proteasome, so you block the degradation, still you can have the recovery of the protein on the surface.
Okay. So another question coming from the States is if you will need other investing partners to develop this project, and how many budget or
this is the very big issue, because we are searching indeed for many money, because now the work from this point on needs a lot of money, because we need to perform the experiment that are so-called high enabling GLP and so on, and this cannot be done in the lab. And so we need million or of dollars or euros to do this. And this is we are promoting our in every part. And by the way, the help of the association, patient association, could be very useful to help us in promoting and to, at the end, to have.
We hope that you succeed. Yeah, yeah.
We are. I have, for example, a meeting next week with the venture capital that should. That is interesting in understanding, in hearing about our story. But of course, you know, the most difficult point is the fact that is a rare disease, and so it's difficult to the perspective of gain at the end is low.
Yeah. Okay, so then a curiosity, but maybe also we can change it in a question, is that from Italy, there is a family that say that she have 2 cases of alpha sarcoglycanopathy in their family and 2 cases of cystic fibrosis that are
Really?
cousins to each other. So then the question is, in your opinion, could be some relationship between these 4 cases, and the same drug will be worthy for all of them, or there are some?
Well,
So you are more or less answered, but
I don't know if I'm able to answer, but I don't think that there is any relationship between the cystic fibrosis and sarcoglycanopathy. You know that cystic fibrosis is quite common in Europe, is frequent, and there is a lot of LT carrier, and so it's quite frequent to have in Italy. If they are in the north part of Italy, it's quite common that they have the disease. Concerning the treatment for cystic fibrosis and sarcoglycanopathy, depends on which kind of mutation they have. If the cystic fibrosis mutation are those that can be rescued by CFTR correctors, because there are a huge number of different mutations, so we don't know. And even though this is the case, it is possible that that compound is not working on sarcoglycanopathy, because indeed it is not the one that we observe to have the best. Of course, we check this molecule, but they are not the best one in sarcoglycanopathy.
Okay, so then from Germany, and this is a general question, is if you have some tips to maintain muscle strength.
Oh, yes, you know, it's really very important for people with sarcoglycanopathy to do some physical exercise. That should be very mild, because of course you don't have to damage the muscle. You cannot do culturismo, how is the okay, but try to move as when possible in a mild way, because this help to preserve the muscle functionality. Yes, this is a
good tip. And then the last one for my part, that is coming from Poland, is asking about the supplement, the HMB, that is the beta hydroxy beta methylbutyrate. I never heard, but I'm looking for. And if they say if they could be useful to combine with this drug or to combine with other treatments, a supplement.
Yes, I know. I think that is a sort of help the status without curing. No, I don't think there is. I think there is no contraindication, but I don't know really how this is working, and so I cannot answer in the proper way.
Okay, so
it's like vitamin or something like that.
It's based on the leucine amino acid.
Okay, see, yes, and so I think that there is no contraindication, but
yeah. And here it's all about the questions that we have received. I don't know if there is some questions in the chat. that to be respond during the presentation?
Yes. Yes.
Okay, so go ahead, Claudia. I close my microphone.
Hello. Hello. Good evening. Can you hear me now?
Yeah.
Can you hear me?
Yes.
Okay. And so I've received two questions actually. And the first one is if this drug is cleared from FDA, will insurance cover this?
The answer, I think, is yes, if it reaches the approval from FDA.
Okay.
I think, yes, I'm not an expert on this yet, I hope. But I think yes, like for cystic fibrosis.
Okay, thank you. And then for a girl affected by LGMD2C, is it possible to have a pharmacological therapy for mutation T521?
T521.
Yes. C.del T521.
It's a deletion. I have to better analyze the mutation, but if it is a deletion that leads to just one or two or two nucleotides that are missing, the pharmacological approach is not possible. If the deletion remove 3 nucleotides, it is possible.
Okay.
It's not sure, but it's possible.
Okay. Thank you so much. Thank you
You're
to
welcome.
everybody. I don't know if there is any other questions. I can't see other questions here. So I think that we can thank Professor Sandonà. Thank you so much. Thank you so much, Carlos. And thank you to everyone who has joined us today. And have a great evening, and thank you again. See you soon.
Thank you.
Bye.
Thank you very much.
Bye.
Have a nice weekend. Thank you very much for give me this opportunity. Thank you very much.
Thank you. Thank you. Very kind of you. Very kind. Goodbye.
Ciao.
See you. Ciao.
Ciao, Carles.
Ciao, ciao.